Till now we have studied about mitosis, which leads to the formation of two daughter cells with identical set of chromosomes. Meiosis, on the other hand, is the cell division in which the chromosome number is halved. For example, if human beings have 46 chromosomes, the end products of meiosis will only have 23 chromosomes. Meiosis leads to the formation of four daughter cells which have haploid genome, which is half the amount of genome which was present in the parent cell. Meiosis produces gametes, for example sperm or egg cells, for sexual reproduction. Meiosis occurs in the reproductive organs or germ cells, for example testes in men and ovaries in women. Formation of gametes generates cells which have genetic diversity, and they assure that after the fusion of male and female gamete, there is a consistent number of chromosomes in the offsprings. That means if 23 chromosomes come from father and 23 chromosomes come from mother, the offspring will have 46 chromosomes. Here you're looking at the male testes. Male testes has certain cells which are germ cells that undergo meiosis to produce sperms. The germ cells have 46 chromosomes, but after meiosis, the sperm only has 23 chromosomes. Before getting into the details of stages of meiosis, let's first understand what are homologous chromosomes. As mentioned before, human beings have 23 pairs of chromosomes. Let's say this is one of the pair that we are talking about. One chromosome came from the mother and one chromosome came from father. Homologous chromosomes have the same type of genes on them at the same locations, but their form might be different. So the two different forms of same gene are called alleles. So you would have one allele here and one allele here at the same location or the same locus of gene. At the time of DNA replication, both the chromosomes in a pair duplicate themselves. These two strands created after DNA replication are completely identical to each other. So these are called sister chromatids, whereas for each other they are non-sister chromatids but homologous chromosomes. The meiosis process starts with the diploid gene that has chromosomes in pair and lead to the formation of haploid cells, which have only half the set or one single set of chromosomes and not the pairs. Just like mitosis, meiosis also has prophase, metaphase, anaphase, telophase, and cytokinesis. But they happen twice. That's why first set of events are marked as 1 and second set of events is marked as 2. Now let's look at what happens in each of these stages of meiosis. A typical prophase leads to the formation or condensation of chromosomes from chromatids, and the chromosomes become visible and pair up. In a pair, you would have homologous chromosomes which have replicated. So this was one chromosome and this is the copy of that chromosome. This is homologous to the first one and this is the copy of the homologous chromosome. Thus, for each other they are sister chromatids, and for the neighboring chromosome it is non-sister chromatid, but as a pair it is a homologous chromosome. So in total there are 23 such pairs in human beings. Where have these homologous chromosomes come from? One has come from the father and the other has come from the mother. But now, to send these genetic information to the child, there has to be a crossover between these chromosomes because we don't want the offsprings to be exactly like their parents. So, there will be a crossing over between the two chromosomes and some genetic information will be exchanged. And these chromosomes will then separate out and lead to genetic diversity in the offsprings. Just like the prophase of mitosis, in meiosis also, the prophase is characterized by nuclear envelope being broken down and start of the formation of spindle fibers. So what are the characteristic features of prophase? One, there will be a tetrad formation. A tetrad would have two homologous chromosomes which have replicated. So you would see 4 such strands and a chiasmata, which is the point of overlap or crossing over between these homologous chromosomes. Now let's look at the events of metaphase 1. Just like the metaphase of mitosis, in meiosis also the chromosomes would start aligning at the equatorial plane. But unlike mitosis, in metaphase 1 of meiosis, you have a pair of homologous chromosomes aligned at the equatorial plane. These two pair of chromosomes assort independently. That means which chromosome will go into which daughter cell is independent of other types of chromosomes getting into that daughter cell. During the metaphase, you will also see the attachment of spindles to the centromere of chromosomes. During the anaphase, the homologous chromosomes are pulled apart and moved towards the poles. But unlike mitosis, here a complete chromosome with its sister chromatid goes to the pole. Therefore, at this stage, if we are talking about human beings, the 46 chromosome pairs will separate out with only 23 getting to one side and 23 getting to other side. During the telophase, the nuclear envelope starts to reappear and it is followed by cytokinesis, which leads to splitting of the cell with two haploid daughter cells. As mentioned earlier, these daughter cells will have only half of the chromosomes, which is 23 chromosomes here and 23 chromosomes on this side. But here you can see, although these are only 23 chromosomes, which is half the number of original, but they are still in the duplicated form. So these duplicated chromosomes will further be separated out into daughter cells. Then begins the second phase of meiosis, which again has prophase, metaphase, anaphase, telophase, and cytokinesis. Now let's see what happens in the second phase of meiosis. Prophase is characterized by condensation of chromosomes. So chromosomes become visible in prophase. Nuclear envelope starts to dissolve because now we have to separate these chromosome pairs into individual sister chromatids. The spindles reappear and attach to the centromere of chromosomes. But unlike the first phase of meiosis, in second phase you do not have crossing over because the crossing over has already happened. But more importantly, crossing over has to happen between the homologous pair of chromosomes. Right now, we already have separated pair of chromosomes. We do not have homologous chromosomes anymore. The single pair of chromosome has separated in one cell, the daughter cell. The metaphase is always characterized by alignment of chromosomes at the equatorial plate. The spindles get attached to the centromere of sister chromatids, and in the next step, these sister chromatids will be pulled towards the poles. In anaphase, the sister chromatids are pulled to the poles, and that is what is visible. So what we have actually separated was this chromosome, one single chromosome that had in the very beginning in the interphase had duplicated itself. But you would see that despite being duplicates of each other, they are not identical. They are not identical because one of the sister chromatid underwent crossing over with the homologous chromosome, whereas the other sister chromatid did not undergo the homologous recombination. So what do we get in telophase? The actual genetic diversity is visible in telophase. We started with these chromosomes, but we ended with these 4 daughter cells, and look at these. They all have different genetic makeup. In telophase, the chromosomes will decondense, the nuclear envelope will reappear, and this will lead to cytokinesis. After cytokinesis, we will have 4 daughter cells with half the number of chromosomes. That means they will be called the haploid cells. We started with the pairs of chromosomes, which is why the cells were called diploid cells, but we ended with half the number of chromosomes, only one set of chromosomes. That is why they are called haploid cells. These haploid cells are called gametes. Meiosis is a very important event for formation of gametes because it gives the opportunity for homologous chromosomes to make crossovers. Homologous chromosomes are able to exchange their genetic material, thus leading to variation in the progeny. But the chromosomes are within themselves assorted independently. Thus, the crossing over of one pair of chromosome does not affect the crossing over or the phenotype or the end product of other chromosome. There will be random exchange of character between homologous chromosomes, there will be random segregation of these chromosomes, and there will be random fertilization of male and female gametes, which is why we have so many chances of genetic variation in a population. Let's compare mitosis and first phase of meiosis. One major difference is mitosis occurs in somatic or non-reproductive cells, and meiosis occurs in the gamete forming cells or reproductive cells. Mitosis begins with duplicated chromosomes. Meiosis begins with homologous pair of chromosomes. During the metaphase, you have one chromosome binding with the spindle, whereas in metaphase 1, you have a pair of chromosomes binding with the spindle. During anaphase, sister chromatids of a chromosome which had duplicated in the interphase are moving apart, whereas during the anaphase 1 of meiosis, the homologous pairs are moving apart towards the poles, so that only one of the chromosomes of a pair get into the daughter cell. There will be the formation of 2 daughter cells in mitosis and 4 daughter cells in meiosis. The 2 daughter cells will have identical genetic makeup in mitosis, but here you can see that all 4 daughter cells have different genetic makeup in meiosis, which is happening because of the exchange of genetic material during homologous recombination or crossing over in meiosis. Let's look at the significance of meiosis. Meiosis gives an opportunity of genetic diversity because it leads to the exchange of genetic material between homologous chromosomes, which come from one from father and one from mother. It's a reductional division. If we start with 46 chromosomes, the gametes will have only 23 chromosomes because each pair has two separate. This kind of cell division leads to the formation of gametes, a very important characteristic feature of sexually reproducing animals. If this halving does not occur, then human beings who have 46 chromosomes will form the gametes with 46 chromosomes. Afterwards, when these gametes fuse, the resulting cell would have 92 chromosomes, and this number will keep increasing generation after generation. Thus, meiosis helps in keeping the chromosomes number consistent in one particular species. There are several chances of getting an error in meiosis. One could be at the stage of separation of homologous chromosomes, and other can be at the stage of separation of sister chromatids. If they are not able to separate properly, this leads to non-disjunction. That means the junction could not be broken between the two homologous chromosomes or between two sister chromatids. Let's look at the situations here. These are the pair of chromosomes, one pair, two pair. These two pairs separate during the first phase, and during the second phase the sister chromatids also separate. Finally, you have 4 daughter cells. But let's say one pair separated properly, but the other pair was not able to separate. Thus, in one cell, you will have a complete set of chromosomes, while in the other, the chromosome pair will be completely absent. This will lead to an abnormal number of chromosomes in the gametes. The other problem could happen when the homologous chromosomes have separated properly, but the sister chromatids have not been able to separate. So you would get 2 normal cells and 2 abnormal cells. There are several conditions or problematic situations that might arise from such non-disjunction condition. For example, when there is trisomy or 3 sister chromatids in chromosome number 21, that is the situation when a child would have Down syndrome, and a situation in which there is only 1 sister chromatid instead of 2, you will have a situation of Turner syndrome in the offspring. So the main takeaways from today's lecture are: meiosis is a reductional division. If you start with 46 chromosomes, it will lead to the daughter cells with 23 chromosomes. It is important for sexual reproduction and evolutionary process because these 23 from male and 23 from female will pair up to form 46 chromosomes or 23 pair of chromosomes in the progeny. And if there are errors in the process of meiosis, there will be error in the number of chromosomes in the gamete, leading to the problem in number of chromosomes or consistency in the number of chromosomes in the offsprings, thus leading to various kinds of syndromes like Turner syndrome or Down syndrome in the offsprings.